Hi, good morning. I am Ananthi, Assistant Professor and Biomedical Engineering Department in Emmanuel JJ College of Engineering. Now I am going to present a topic on Introduction to Biochemistry. So now let's move on to the topic. First, initially, before moving to the topic, the structure of inside this introduction to biochemistry, we have many topics inside that. In this video, we are going to cover all the topics inside that. First, initially, we are going to the topic structure of water and its significance. Basically, if we take the basic water molecule, we all know that water molecule is composed of two hydrogen atoms and one oxygen atoms. These atoms are held together by covalent bonds. The electron pairs are shared between oxygen and hydrogen. These are the common. Every hydrogen atom forms a single bond with a central oxygen atom. The arrangement of these atoms is not linear. Instead, the water molecule has a bent or V shape. Next, continuation of that slide, that oxygen atom is more electronegative than hydrogen, which means that it has stronger pull on the shared electrons. This unequal sharing causes the oxygen atom to acquire a slight negative charge, while hydrogen atoms develop a slight positive charge. This makes water a polar molecule with distinct positive and negative ends. This is the basic water molecule hydrogen bond. Inside that, many bonds are interrelated. In between hydrogen center, we have an oxygen. The two sides have hydrogen bonds which are interconnected by bonds. The hydrogen have a positive charge and the oxygen has a negative charge. This is the electrical attraction between the water molecules. This is the basic structure, how the positive and negative ions are interconnected. Let's now move on to the topic, physical properties of water. In chemical formula, first we take the chemical formula. Water, that is H2O, is the chemical formula for water. That we all know that. The covalent bonds exist between the hydrogen and oxygen atoms in the water molecules. A single atom of oxygen forms a connection with two hydrogen atoms. The appearance is, in its natural condition, water is a colorless, odorless and tasteless liquid. The boiling point, when we take the boiling point, water has a boiling point of 100 degrees Celsius. As we all know that 100 degrees Celsius is the tip boiling point of the maximum boiling point of the water. However, water's comparatively high boiling point violates the periodic table tendency. The hydrogen net hydride has a relative high boiling point when compared to hydrogen telluride and hydrogen sulfide, both of which has low boiling points. This is due to the extremely strong hydrogen bonds in the water molecule. It takes a lot of energy for them to break and start boiling. Next, when we take the freezing point, the same notation applies of water freezing point. Water has a freezing point of 0 degrees Celsius. When we compare with the melting point is the 100 degrees Celsius and the freezing point is 0 degrees Celsius. The fact in this water, it takes a long time to freeze or even boil also. Is critical for our ecosystem and its existence. And when we take the density, one of the remarkable points of water is that it is dense in its solid condition. The density of water increases as it cools up to 4 degrees Celsius. However, after that, water gets less dense. Ice floats in the water. When we take the ice, it will floats in this water. Because of the density of water increases as it cools up to 4 degrees Celsius. Due to this reason only, ice water floats in the water. Then when we move to the topic viscosity, due to very strong intermolecular interactions, water has a high viscosity. Solvency, when we take the solvency, water is a highly effective solvent. It is even referred to as universal solvent. Then the chemical properties of water. When we take the chemical properties of the water, its atmospheric nature. First take the topic atmospheric nature. Water is atmospheric in nature because it can act as both acid and base. So it is an atmospheric nature. Following two reactions show that the water is a atmospheric in nature. That is, when H2O is added with hydrochloric acid, it gives H3O plus Cl minus. When water is added with nitric acid, it gives the two sides of action, NH4 and OH minus. Redox reactions. Water is reduced to a hydrogen molecule by electropositive elements. As a result, water is a rich source of hydrogen. Water is oxidized to O2. During the photosynthesis process, water is very useful in redox reactions because it can be oxidized and reduced. When we take the hydrolysis reaction, because of its dielectric constant, water has a very strong hydrating tendency. It dissolves a wide range of ionic compounds. Water can hydrolyze some covalent and ionic bonds. This is the differentiation of covalent and non-covalent bonds. Different properties are distinguished here. That is, attribute. When we take the attribute for different attributes are discussed here for covalent bonds and non-covalent bonds. First, when we compare the formation for covalent bonds, it's sharing of electrons between atoms. Non-covalent bonds, weak electrostatic interactions between molecules or ions. When we take the strength, covalent bonds has strong strength, but for non-covalent bonds it has weak strength. When we take the energy, high energy required for bond formation, and for non-covalent bonds, lower energy required for bond formation. When we take the polarity for covalent bonds, it can be polar or non-polar. When we take the non-covalent bonds polarity, it can be polar or non-polar. Both sides are having the same polarity. Examples of covalent bonds: carbon-carbon bonds, oxygen-hydrogen bonds, and for non-covalent bonds, the examples are hydrogen bonds and van der Waals interactions. Next, covalent bonds and non-covalent bonds are the two types of chemical bonds that holds atoms together in molecules or compounds. Covalent bonds involve the sharing of electrons between atoms that resulting in a strong and stable bond. These bonds are typically found in molecules with two or more non-metal atoms. On the other hand, non-covalent bonds are weaker and involve electrostatic interactions between atoms or molecules. These bonds include hydrogen bonds, ionic bonds, and van der Waals forces. Non-covalent bonds are important for maintaining the structure and function of biological molecules such as proteins and DNA. While covalent bonds are stronger and more stable, non-covalent bonds play crucial roles in various biological processes and interactions. First, let's move on to the covalent bonds. In covalent bonds, it occurs when two atoms share electrons to achieve a stable electron configuration. This sharing of electrons creates a strong bond between the atoms that resulting in the formation of molecules. Covalent bonds are typically formed between non-metal atoms such as carbon, oxygen, and nitrogen. These bonds are characterized by their directional nature, meaning that shared electrons are localized between the bonded atoms. Then, covalent bonds also exhibit a wide range of bond lengths and bond energies. The bond length refers to the distance between the nuclei of the bonded atoms, while bond energy represents the amount of energy required to break the bond. Different atoms or molecules having varying bond lengths and energies depending on factors such as the number of shared electrons and the electronegativity of the atoms are involved. Then, Covalent bonds, actually the covalent bonds are essential for the formation of organic compounds. That organic compounds are the basic building blocks of life. Organic molecules means such as proteins, carbohydrates and nucleic acids that rely on covalent bonds to maintain their structure and function. The ability of the covalent bonds to form stable and specific connections between atoms allows for the complexity and diversity of biological systems. Next, we move on to the topic non-covalent bonds. These non-covalent bonds, also known as weak chemical bonds, are the interactions between molecules or parts of molecules that do not involve the sharing of electrons. Unlike covalent bonds and non-covalent bonds are relatively weaker and more transient in nature. They play a crucial role in various biological processes such as protein folding, DNA replication, and cell signaling. One of the primary types of non-covalent bonds is the hydrogen bond. That is, hydrogen bond occurs when the hydrogen atom covalently bonded to an electronegative atom that interacts with another electronegative atom. This interaction leads to the formation of the partial positive charge on the hydrogen atom and a partial negative charge on the electronegative atom. Hydrogen bonds are responsible for the unique properties of water such as its high boiling point and surface tension. Van der Waals forces are the another category of non-covalent bonds. These forces arise from temporary fluctuations in electron distribution that leading to the formation of weak attractions between atoms or molecules. Van der Waals forces can be further divided into three types. That is London dispersion forces, dipole-dipole interactions, dipole-induced dipole interactions. These forces are responsible for the interactions between non-polar molecules as well as the stability of molecular structures. Non-covalent bonds are crucial for the three-dimensional structure and function of biomolecules. For example, proteins fold into specific shapes due to the interactions between different amino acid residues, which are primarily mediated by non-covalent bonds. Additionally, DNA molecules are stabilized by hydrogen bonds between complementary base pairs, allowing for the accurate transmission of genetic information. This is the acid-base maintenance slide. Acid-base balance means it is measured using the pH scale. A variety of buffering systems permits blood and other bodily fluids to maintain a narrow pH range, even in the face of perturbations. A buffer is a chemical system that prevents a radical change in fluid pH by dampening the change in hydrogen ion concentration in case of excess acid or base. Most commonly, the substance that absorbs the ions is either a weak acid which takes up hydroxyl ions or a weak base which takes up hydrogen ions. Buffer systems in the body, buffer systems in the human body are extremely efficient, and different systems works at the different rate. It takes only seconds for the chemical buffers in the blood to make adjustments to pH. The respiratory tract can adjust the blood pH upward in minutes by exhaling CO2 from the body. The renal system can also adjust blood pH through the excretion of hydrogen ions and the conservation of bicarbonate, but this process takes hours to days. It has an effect. The buffer systems functioning in blood plasma, that includes plasma proteins, phosphate and bicarbonate and carbonic acid buffers. The kidneys help control acid-base balance by excreting hydrogen ions and generating bicarbonate that helps maintain blood plasma pH within a normal range. Protein buffer systems work predominantly inside the cells. This is the acid-base balance, that is homeostasis, is disturbed. That pH downwards, pH upwards, it is classified into acidosis. When the pH range comes below the range, it is acidosis. When the pH range goes outside above the range, it is called alkalosis. That acidosis stimulates brain and arterial receptors. Alkalosis stimulates brain and arterial receptors. That is, respiration rate is high, alkalosis respiration rate is low, blood carbon dioxide is low for acidosis and high for alkalosis. Blood carbonate H2CO3 is low for acidosis and high for alkalosis, and the pH range is high for acidosis and low for alkalosis. Then it is gives to restored. The two are combined together to the restored state. This is the flow chart, which can, in this way, we can easily understand the two processes in acid-base homeostasis. That the two processes are acidosis, alkalosis. Acidosis means the pH range becomes low. Alkalosis, pH range is low. And when we take this side acidosis, which is low, higher means here this side low. Opposite signs will occur. If we understand the one concept clearly, the other concept will get clear by using flowchart. Then what is meant by this homeostasis? The human body is designed to self-regulate biological processes and maintain internal stability. A healthy body has better maintained its balance when it is in a state of homeostasis. That is, when our body is healthy, it is in the state of homeostasis, and the cells and organs of the body function properly. That is, our body is stable, so our organs and the cells in the body all will work properly during the stage of homeostasis. It maintains ideal body temperature, pressure, pH, sugar level, reaction rates, and many other vital biological processes. As part of homeostasis, several buffer systems operate cooperatively in the body to maintain health. A buffer system is essentially composed of two components: main component and its counterpart component, each of which must be present at an appropriate balance and threshold to ensure more effective buffering. In the body's aqueous solution, certain chemicals can behave in two ways, as hydrogen ion donors and acceptors. In that similar way, we have two types of neurotransmitters, excitatory and inhibitory, two sources of energy, potential energy and entropy, and two metabolic processes, anabolic and catabolic. At the personal level, values can take two common forms, material and non-material. Behavior can take two modes, permitted and prohibited. It is appropriate to evaluate those potential buffering systems by analogy to acid-base buffers, and the results are interpreted in terms of maintaining balance and health. This is the diagram in homeostasis. Initially the body is stable. When in the state of homeostasis, it is given in the triangle shape, and when it goes to buffering state, it gets slightly deviated. Without buffering, then it gives to that slide. It is a circulatory process. When it buffering state, it slides, and without buffer, it takes to opposite state. When then buffering is given, then it comes to again triangle shape. By seeing this diagram, we can get an idea how buffering minus and cross symbol is given. Positive and negative charges, like with buffer, without buffering, are clearly explained using this slide. Then Henderson-Hasselbalch equation. This Henderson-Hasselbalch equation indicates that pH is determined by this ratio. That's pK effective, the value is 6.3. This gives the ratio of 0.08, that is 8 by 100. There is effectively 12 to 13 times as much carbonate aqueous as CO2, making the system primed to react with acid produced metabolically. Yet, the second conundrum exists. The pH of a blood, 7.4, is outside the optimal range for a buffer system. In this case, plus or minus 1 pH unit from the pKa, again the system is primed to react with acid, as this would move the pH closer to the optimal buffering pH of 6.3. Other biological system must also involved in maintaining the pH. Then, the respiratory alkalosis can be caused by hyperventilation or breathing rapidly. This can lead to breathing out, that is, removing too much CO2, shifting the above equilibrium to the left, consuming H3O plus and increasing pH, making the blood more alkaline. To increase your CO2 levels, you could breathe into a bag. Respiratory acidosis is caused by increased CO2, which can occur when the lungs are in working well and you can't get rid of CO2 you produce during respiration. Respiratory acidosis can occur with asthma, pneumonia, lung disease, or any condition that decreases the respiratory rate. Then, next we are moving to the topic viscosity. Viscosity, it is the resistance to the flow of a liquid. That is, low viscosity liquid will fill the tumbler. That is, for example, water, it's a low viscosity. That will fill the beaker faster. But it is when we look to that beaker, that low viscosity liquid, water is filled slowly only, but the beaker is filling faster when compared to high viscosity liquid. High viscosity means, example, honey. honey filling the beaker very slowly, but the water filling the beaker very fastly. It is comparedly given, so we can easily guess the viscosity and low viscosity and high viscosity liquids. Low viscosity example water, and high viscosity example honey. Then this is the viscosity formula and the tabulation. It is given versus displacement versus velocity. That the displacement is given by y, that is in the x-axis we are given velocity. It is plotted against velocity versus displacement. X-axis velocity, it is given in meter per seconds, and the y-axis displacement is given. It is given in meter. That is fluid that viscous is increasing. That is given by shear force. That area that is, it is given by velocity by gradient dou u by dou y. Normally the formula is given by viscosity formula, viscosity symbol eta. Eta is equal to F by A into dou u by dou y. Then some of the factors that are affecting the viscosity, that the viscosity of liquid decreases quickly with an increase in temperature. On the other hand, the viscosity of gases increases with an increase in temperature. The liquids flow more smoothly upon heating, and gases flow more sluggishly. That is sluggishly, what is the meaning is, that is dim light. Without any activeness, it is low. The viscosity of gases is approximately proportional to the square root of temperature. The reason why gases increase their viscosity is because of an increased frequency of collisions. The more the molecules collide, the disorganized they become. As a result, they move sluggishly, that is without any activeness, that is like a dim light. It sluggishly goes. It is the graph plotted versus velocity versus displacement, that shear stresses vary, how it is flows, and the gradient is given by dou u by dou y. And then surface tension. The surface tension is the tendency of the liquid surfaces to shrink to the minimum surface area due to the cohesive forces. That is, when we look to the diagram, due to the molecules are pulled from all directions. Surface molecules are pulled inwards. That is, inside that molecules are pulled in all directions, but the surface having molecules are pulled only during downward direction. That is inward. It is not pulled outwards. In the symbols and units of surface tension, the most common symbol for surface tension is gamma. That is lowercase Greek letter gamma. SI unit is Newtons per meter. Per CGS unit is dyne per centimeter, dynamics per centimeter. Other includes some other units are also added here. That is F per centimeter square and J per meter square. Next, the examples of surface tension. First example, the formation of water droplets. That is, raindrops adopts a spherical shape to minimize surface area. Floating needles, the steel needle rests on the water as surface tension prevents it from sinking. Then capillary action, water rises in a narrow tube due to cohesive and adhesive forces. Soap bubbles, the spherical shape of bubbles results from surface tension acting uniformly. Walking water striders, some insects, when we look in the water, some insects can exploit surface tension that tends to walk on water. Meniscus formation, concave or convex surfaces from where water meets a solid boundary. Liquid jets breaking, a falling stream of water fragments into droplets due to surface tension. Tears of wine, thin liquid films form due to differences in alcohol concentration. Spreading of oil, A thin film of oil spreads on water that reducing surface tension. And when we see to the medical side, lung function, surface tension in alveoli is critical for breathing mechanics. It is regulated by surfactants. Some of the formulas for surface tension, the basic formula is gamma is equal to F by L, where gamma is equal to surface tension newtons per meter, F is equal to force that is given in Newton, and L is the length over which force acts, it is given in meter. And capillary rise method, it is given by gamma is equal to H rho gr by 2, where H is equal to height of liquid column, it is given in meter. Rho is the density of the liquid, it is given in kilogram per meter cube. G is the gravitational acceleration, it is given in meter per second square. R is the radius of the capillary tube, it is given in meter. And next we are going to the topic adsorption. An adsorption is a process in which molecules or particles adhere to the surface of the solid or liquid substance without penetrating its structure. This adherence occurs due to attractive forces between the adsorbate surface substance being adsorbed and the adsorbent surface on which adsorption occurs. So, in this topic we will discuss what are the characteristics of adsorption and types of adsorption. The adsorption and absorption, the two, when we saying this point, when we hears it, the sounds look similar. What's the difference? Let's see. Absorption means this process involves one substance being taken in and fully incorporated into another substance. And adsorption, in this process of adsorption, molecules of a substance adhere or stick to the surface of another substance without penetrating or being adsorbed into its bulk. In adsorption, molecules of one substance penetrate into the inner structure or volume of another substance. And in adsorption, it involves the accumulation of molecules or particles on the surface of the liquid or solid or liquid. For instance, a sponge absorbing water, it involves the water molecules being soaked up and distributed within the sponge's material. And for adsorption, an example, when gases or solutes adhere to the surface of a solid, like how activated charcoal adsorbs impurities from the solution. Then diffusion and osmosis. It has the same concept only past. For osmosis, it has solution and pure water in between the semi-permeable membrane. And diffusion, high concentration liquids moves to the low concentration area. Here in between we have a semi-permeable thing. In that, pure water passes through the semi-permeable thing, and solution, it is not passes through the semi-permeable membrane. Only the pure water is transformed through it. Then diffusion. Diffusion is a fundamental physical process characterized by the spontaneous movement of molecules or particles from an area of higher concentration to an area of lower concentration. This movement occurs due to the intrinsic kinetic energy of particles, leading them to disperse and spread out in a continuous effort to achieve a state of equilibrium. Diffusion is omnipresent in various natural phenomena, playing a crucial role in processes ranging from the exchange of gases in biological systems to the blending of substances in liquids and gases. The principles of diffusion are encapsulated by mathematical models such as Fick's laws, providing a quantitative understanding of the factors influencing the rate of diffusion in different mediums and environments. We have two laws here, first law of diffusion and second law of diffusion. In first law of diffusion, it is given by J is equal to minus d dc by dx, in which J represents the rate of diffusion, D stands for the diffusion coefficient, a property dependent on the nature of the diffusing substance and the medium. dc by dx, it denotes the concentration gradient, signifying the change in concentration per unit distance. And in second law of diffusion, it is given by J is equal to minus dA dc by dx. Here J is equal to minus d dc by dx. Here on A is added. That A represents the cross-sectional area. A represents the cross-sectional area through which diffusion occurs. The diffusion in cellular processes or gas exchange in lungs, that is oxygen and carbon dioxide exchange between air and blood. Nutrient uptake in the cells, that the cell will absorb the nutrients from the extracellular fluid. Next, waste elimination, that is the removal of waste products from the cells. Next, osmosis. Next topic, osmosis. That is, what is meant by this osmosis? This osmosis, it is a fundamental biological process that defined as the spontaneous movement of solvent molecules through a selectively permeable membrane. This movement occurs from a region of high water potential characterized by lower solute concentration to a region of low water potential where solute concentration is higher. The ultimate goal of osmosis is to equalize solute concentration on both sides of the membrane, facilitating equilibrium. This phenomenon, is essential for maintaining homeostasis in biological systems. That is, so we have seen about here first we see about viscosity, some of the factors in this, how introduction to biochemistry we are seeing in the first unit. This viscosity, surface tension, osmosis, diffusion, these all things are studied due to how it is used in the biological, or how it is gets affected in the biological system only. To know that only, here all the separate topics are discussed here. That is, viscosity means in low density liquid will flow faster in the beaker, and high density, for example honey, it will fill the beaker slowly. And the formula is given, and how the shear stress is given here. When we see this graph, the shear stress will be understand to ourselves very clear. And the velocity gradient formula is given it is given when we see that it is given in the shape of right triangle, that is dou u by dou y, and the velocity is given mu is equal to meter per seconds. And also some of the factors how viscosity is affected, that is, we all seen that liquid smoothly heat upon heating when compared with heated liquids and gaseous flow. The gaseous flow will suggestly, that is, it will not be active. That is, when we see bright light and dim light, we will see the difference. Like a dim light only, that the meaning of sluggishly. sluggishly the gases will flow. The viscosity gases is approximately proportional to the square root of temperature. Next we see, already saw about this, that same formula is given here, gradient, by using a graph how it is expanded in the shear stress. It is given by tau, that is the expanded region is given by shear strength. The expanded region only mentioned here as shear stress, and how velocity displacement in the y column. Then surface tension. Surface tension, when we take a molecule, sorry, um, liquid in a beaker. In between the liquids are inside that, how many molecules are present. In the middle side, when we see the liquid, in that concentration between that liquids are moves in all directions. If we place a point in the middle of the liquid in any molecule, in that molecule the movement is all side, downward, upward, left side, right side, everywhere. But when we take the upper side, that is surface side, having the molecule waters, that will move only downward, leftward, rightward, not upward. That is clearly explained in this diagram. That forces between two molecules is known as cohesion. Cohesive forces is played in this between the liquids. That every forces has attractive forces, that two molecules between what forces present means, that is known as cohesive force. That is, the surface tension is the tendency of the liquid surfaces to shrink to the minimum surface area due to cohesive forces. That bulk molecules are pulled out from all directions, and the surface molecules are pulled inward. Some of the formulas which are symbols and units which are used in surface tension are Dow newtons per millimeter is the SI unit. Some other units are also here explained here. Some of the examples of surface tension: formation of water droplets, floating needles, capillary action, soap bubbles, working water striders, meniscus formation, liquid jets breaking, tears of wine, spreading of oil, lung function, etc. are given in this examples of surface tension. Next formula is Dow is equal to F by L. Gamma is equal to F by L. Gamma is our symbol for surface tension, that is given in newtons per meter. F is the force, that is Newton. L is the length over which force acts, that is given in meter. Then also it has another one formula, capillary rise method, that is gamma is equal to H rho gr by 2. H is the height of the liquid column, it is given in meter. Rho is the density of the liquid, it is given in kilogram per meter cube. And g is the gravitational acceleration, it is given in meter per second square. R is the radius of the capillary tube, it is given in meter. And adsorption. Next, adsorption. It is the formation of molecules or particles adhered to the surface of the solid or a liquid substance without penetrating its structure. Difference is, adsorption means the process involves one substance being taken in and fully incorporated into another substance. But in the process of adsorption, molecules of a substance adhere or stick to the surface of another substance without penetrating or being absorbed into its bulk. This is the difference between osmosis and diffusion. In osmosis, in between pure water and solution, we have a semi-permeable membrane, but in diffusion, the low concentration molecules will move to the higher concentration area. So these are the topics we saw here. Then diffusion is also given. Next, first law of diffusion formula. We have second law of diffusion formula. The differences we have in minus d dc by dx. Second law, we are initially area is given. Then different cellular processes are given. Next, osmosis. What is meant by osmosis is also explained here. Already I explained. So these are the topics in Introduction to Biochemistry. Thank you.